All right, welcome to the Center for Reproducible Biomedical Modeling seminar series. So in this series, we invite speakers to discuss different tools and methodologies that address various issues that come up in biomedical modeling and reproducibility. In the summer series, there have been two talks. The first was on June 28th. That was with Dr. Pedro Mendez, who discussed fair data principles as applied to biological models. If you missed that, I suggest go back and checking it out. That video should be on the website soon. Also check out previous seminars. Today we have Paul Jost. He's going to discuss pyPESTO, a platform for parameter estimation. The format of these talks is 15-minute presentation, 10-minute group discussion at the end. Feel free to post any questions you have in chat, and we'll try to collect those if you'd like. A little bit about Paul. He's a PhD candidate at the University of Bonn, where he studies lipidomics and single-cell lipid metabolism. To facilitate those studies, he develops different tools such as pyPESTO for parameter estimation. A side note, we had the pleasure of hosting Paul in the SAR lab this March, where he and Lucian integrated Roadrunner into pyPESTO. So if that's interesting to you, go ahead and check that out. And with that, I will hand it off to Paul. It's all yours.
Thank you, Michael, for this kind introduction. I need to be able to share my screen. I'm currently not allowed to.
That should work.
Yes. So, always the important questions. Can you see my screen?
Yes.
Nice. Then the second important question, can you see my mouse? Because also sometimes happens.
Yes.
Okay, good. Then we are ready to start. Okay, yeah. Thank you again, Michael, for this kind introduction, and welcome everyone to today's talk on pyPESTO, a modular and scalable tool for parameter estimation. Usually people have a thank you slide at the very end. I do have that as well, but as it is with tools that are written in a large manner, they don't survive by just having one person there. And that's why I think I need to thank already here at the very start all the pyPESTO developers, people that contributed to it, and people that help keeping the tool alive. Okay, and with this we can start. What is today's talk going to be about? It's going to be about pyPESTO. pyPESTO stands for Parameter Estimation Toolbox for Python. As the name suggests, it is a Python toolbox and a Python program. So if you use C++ only, I think we can't help you there yet. Might be at some point. And it is used for parameter estimation. Why do we call it a toolbox? Well, as it will turn out in a few slides where I introduce also the parameter estimation is not just one step, be done with it, but it consists of a multitude of steps, and each step has to be done multiple times. And with each steps, you have the choice of a variety of tools that you want to use. And just using them raw would mean that you have to transform your data, your code, in a manner that suits the corresponding tool. And this is where pyPESTO comes into play. It helps you streamline your code, helps you streamline the whole parameter estimation by providing interfaces to many tools that you can use within the parameter estimation pipeline. The structure of today's talk is going to be that first, to get everyone on the same page, I'm going to talk about how do we get from a reaction to a mathematical model. Then I'm going to try to convince you that parameter estimation is really a crucial part of designing a mathematical model. And then from there on out, we can take a look at how does parameter estimation look like? How do we find a parameter estimation problem? and how does pyPESTO aid you in each step of parameter estimation. My very last slide will be my last try to convince you that pyPESTO streamlines parameter estimation. And I think we can start. Okay. So from reactions to mathematical model, I chose a very simple biological application that always baffled me when I first stumbled over it. It is the Valkov oscillator. It's a very simple description to this for glycolysis, and it only consists of three reactions or three things. It assumes a constant influx of ATP, which henceforth we'll call S or substrate. Then ADP gets converted to ATP, which we'll call product or P. And then ATP gets converted to something else, transported. We don't really care in this case about it, and so we say it enters the sync state. So the first thing you do is you have the description and you want to convert this into some kind of reaction. For this, you need to assume some kind of reaction kinetics. There's a multitude of them again. There's Michaelis-Menten reactions, and there's, in our case, what we'll be using, mass action kinetics. So the first reaction is quite straightforward. It says from nothing there shall be substrate at a constant rate. So we should just say, well, the flux will be some kind of constant parameter K1. The second one is a bit more involved, and where the mass action comes into play, basically says one substrate reacts with two products to three products, basically converting the substrate to the product. And mass action assumes that the more of a concentration of the reactants is there, the more often a reaction happens. So the reaction flux will be some constant, we don't know this yet, K2, times the concentration of S and P squared. The third one, again, we enter a sync state, so P goes to nothing. And the more P there is, the more it enters the sync state, so it's K3 times P. So if the reactions, this is not yet the mathematical model, and for the mathematical model, we have a multitude of options. This is by no means exhaustive. It's just the first three I came up with. The first one is you just assume, well, each reaction has a certain probability to happen or to occur. So we want to do a stochastic simulation, like a Gillespie algorithm. You might want to say, I want to have a spatial component in there and model the concentrations explicitly. Then you have a partial differential equation. Or you say, I assume the concentrations are fairly well mixed and the reactions happen so often, it's basically deterministic. You might want to use an ordinary differential equation. We will stick to ordinary differential equations also throughout the whole talk. But just keep in mind, there are other options, and you could even use other options together with pyPESTO. For the ordinary differential equation, we describe the changes in time of the concentrations here by just multiplying the flux vector with the stoichiometric coefficient. In short, stoichiometric coefficient means for each reaction, how much of the substrate and how much of the product was used and generated. So it's a matrix of, in this case, 0 and 1s, or minus 1s. If we multiply this together, we get two equations for both of them, and we are basically happy already to describe the dynamics. The only thing we need is we need a starting point. And so we say, okay, the starting point is also, again, some unknown parameter. So what we have in total now is two equations, three reactions, and 5 parameters. 3 of those are reaction parameters, 2 of those are initial state parameters. We don't know. Fairly simple, fairly straightforward. But what if I told you that out of those two equations, 3 reactions, 5 parameters, I can, by changing only the parameters, not the reactions, not the equations, get 3 completely different qualitative behaviors. In the middle, we have what gives it the name, the Valkov oscillator. In this case, the model will really go up and down in concentrations. They will interchange, and it will actually enter a stable cycle in the end. The other ones are no official names. I came up with them myself. in the first case, the growth oscillator means that if we change the parameters as we want to, then we can have the substrate just explode. It will grow infinitely or indefinitely. Yes. And there's a set of no oscillator, which means we don't have an oscillating system, but we actually enter a steady state. We keep it stable and it will stay there as long as we wait. You might say, okay, you're changing 5 parameters in 2 equations. That's already a lot. But what if I told you that we reach those 3 different behaviors by only changing exactly 1 parameter? We kept all the other 4 parameters constant and only changed K2 from 1 to 20. To me, this was very surprising. And I think what it tells you is parameter estimation is crucial to determine dynamics. It's not just enough to say, okay, well, I have the model structure, and now I can start looking into how the behavior looks, but we really want to pinpoint the parameters to a precise value. Even if you know all the others by some kind of literature knowledge already, you still want to pinpoint the last parameter. So that means, jumped over this, there is a necessity to precisely estimate parameters using some kind of real world data or some kind of measurements, because we want to see how does it behave in reality. Okay, so how do we combine model and data to get to our parameter estimation? We do this by extending our model. Again, there are a multitude of approaches. The one that pyPESTO supports quite strongly, and which makes your life easier, is the likelihood estimation. You extend the model, the first things you know is we already talked about the vector field and the initial values, meaning that we describe the changes as some kind of function that is dependent on the current state, theta are the unknown parameters, and u are some kind of known parameters. The initial values are the same. And we now introduce observables. Observables are basically, what do we measure? And they are described as a function of the state at a given time and some additional parameters. How does this look like? In the easiest case, we had this example of ATP, ADP. We can directly measure ATP concentration. In this case, the H function would be just an identity. This is usually not the case. Usually you measure some kind of scale data, for example. In the best case, there would be some kind of parameter times the ATP. What is also usually a scenario is we have multiple compartments in a cell, but when measuring, we only measure the total amount. And then H is some kind of sum over all the ATP in the different compartments. So there's a multitude of options. It's just defining what do we measure. It is highly dependent on the measurement process. This is just the theoretical function of what we measure. What we actually measure is this function plus always some kind of error. Independent of how we measure, there will always be something that we can't control. It's like temperature, could be air pressure, can be any kind of things that change the values in ever so slight manners, and we describe this by some kind of error. The error distribution is completely up to you, but what normally is used is some kind of additional normal distribution, the multiplicative log normal distribution. You're free to choose. And from there, what we can do is we can take a parameter set, which we don't know. We simulate the model, take our observables, and then subtract them here. This leaves us only with the error, which is normally distributed. So we can create a density function. Then if we assume that the error is independent, we get the density function over all the data. And this is the likelihood function. And what we now do is we maximize this likelihood function, creating the parameter that, given this parameter, it is most likely to have produced the data. So no other parameter is more likely to produce the data. It's still a bit different than the posterior, which we'll perhaps talk about today. I'm not sure, I don't think so. Okay, so we have a function that we want to optimize. So how does parameter estimation look like? Parameter estimation to me is something like a never-ending story, honestly. We talked about model equations and experimental data, and the first step, we are kind of halfway there already. we'll get more a bit into the details of formulating an objective function. This includes, if you don't want to write it yourself, choosing a simulator like Roadrunner. If you did this, you can theoretically directly start with the parameter optimization. And this is where the first choice you have to do comes in as well. You have to choose the optimizer. There's a multitude of options for the optimizers. You can choose local optimizers, global optimizers, stochastic optimizers, all of them. And each optimizer has meta parameters, again, some kind of options that you also can fine tune. If you chose them, you optimize, you get some kind of parameter estimates. And then you come to the most crucial point of parameter estimation. As simple as it sounds is, how well does your data fit? Because the parameter estimation can look as good as it is. The parameters can be as interesting as they are. If they don't fit the data, they don't have a real value for this parameter estimation problem. But then you need to decide, where do we want to go back to? And there come some additional things into play. As I said already, there's a multitude of options. But perhaps you always stopped too early with your optimization, and so you want to make more stringent convergence estimates. Or you can say, well, one of my parameters is always hitting a bound, so I need to change the bounds of my parameters, so the maximum values I allow for the parameter and the estimation. You'd go back to formulating the objective. In the worst case, but it's also a good case, both at the same time, you can't describe the data in any way, shape, or form with the model you have. That means if it's, for example, a well-established model, something is wrong there, something is missing. This is really nice. It usually puts you in some kind of unknown field, and then you are also in the model selection part. I'll talk about this later. Let's assume our model fits really well. We can continue. We've now found a parameter that describes the data, but how sure are we that this is actually the real parameter? This is where practical identifiability analysis comes into play. Again, we have the choice of multiple methods, and what we want to obtain from them is basically some kind of confidence intervals on the parameters, telling us, this parameter, we are fairly certain it's in this range. For this parameter, we are really not so sure. It never ends with the parameters, because what you're usually interested in is the output of the whole model, the simulation. And so you want to somehow propagate the uncertainty in your parameters to uncertainty in the prediction, which is the fifth step, the prediction uncertainty. Again, here's the question, how do we propagate this? Both of those together show you uncertainties. And if the uncertainties are too high, you cannot dodge the bullet, and you have to, in some way, shape, or form, redo experiments. Either you just need more data, that's the best case. You might need some different data, a different experiment, a different observable, anything like this, and you're back to square one. And that's where it starts again, and you might have to do it again. As I said, an almost never-ending sword. Something, if you ever see a light at the end of the tunnel, basically, you say, okay, well, we are a calibrated model. Everything is fine. Everything is identifiable. That still doesn't tell you that it's the only model that is identifiable or that describes the data well. So you might want to do some kind of model selection, looking what is the best model for this. And again, here, you have to choose some criteria of what is best. You choose a method of how to exactly check for the best value. Now describe to you what parameter estimation looks like. We'll first now do, how do we define a parameter estimation problem in a hopefully reproducible manner, because we're at the center of reproducible biomedical modeling. And then we'll talk about how does pyPESTO aid you in each of those steps. Okay, for creating a parameter estimation problem, I want to introduce you to PETUP. PETUP is a data format for specifying parameter estimation problems, and it is a standardized format that is used to share parameter estimation through our toolboxes. Because what you really want to do, basically, is I take my parameter estimation problem from one toolbox, tell the other toolbox, here, this is it. Here you have some files, please do something with it, and then hopefully, without having to do anything at all, you can use the other tool for the parameter estimation. It is built around SBML, and tabular separated files. How does this look like? We think of the most common setup we can imagine in a parameter estimation. So first, we already talked about a generic model. This is encoded in an SPML model. We also talked about the observables. I also clicked the conditions, I'm sorry. We talked about the observables. You put them also in a table with the noise distribution and the noise parameter. For the data that you have, you think of every kind of data set as an experiment and some sort of condition, meaning that the conditions just vary between the experiments. Either you knock out a reaction, you know that you start in steady state, or you know that one of the initial conditions is perhaps 0 or something. And then for each of those, someone clicking double, I'm not sure why, perhaps it's late. So for each of those experiments, you have some kind of data. Within all those tables and the model, we accumulated parameters. Here are some scaling parameters. Those might be some parameters we want to estimate. And obviously in the SPML model, there are parameters. So this doesn't really work. Yeah. So the last thing, probably the most crucial part for parameter estimation, is the parameter table, where you list all the parameters and you say, do I want to estimate them or do I not want to estimate them? Reasons to not estimate them is you have prior knowledge. And you know, okay, we can fix this value there. You can also have prior knowledge on the distribution of the parameters. This is a bit more involved. Okay, so first step, let's only press one button at a time. We are at the objective function definition. How does pyPESTO do this? Well, pyPESTO interfaces a variety of simulators, but just only saying this once, because I hope no one really wants to do this. pyPESTO is able to also work with your self-defined objective functions. All it really needs is an evaluable objective function. So you can define the whole likelihood yourself. You can define a simulator yourself and do everything. I highly advise against it because it's really a hell of a lot of work. If you decide against it, it is really easy to set up the problem because through the support of PETAB. So PETAB basically tells you, okay, we want to do a likelihood estimation and you need a simulator. And pyPESTO supports a multitude of simulators. One of them is Amici. Another one recently added is Roadrunner. You can define your own models in Julia through the support of PETAB Julia, for example, which is also a Julia package, or you can use JAX for faster gradients. If you do it like this, the whole probably 1,000 lines of code, if you do it raw, would be boiled down to 3 lines. We load the PETAB problem. We create an importer that is able to create the problem. This one here, for example, tells us it wants to use Amici. If we slightly change the behavior, it tells us it wants to do Roadrunner. And then you just say, well, create the problem, be done with it. Now we have a problem. That means we have an objective function we can evaluate. We know parameters that we want to estimate and can change them if we so want to. And with parameter bounds, now saying within these bounds, the parameters should be found. Okay, next step, choosing amongst a variety of optimizers. So as I said before, there's a multitude of optimizers. And the sad message is there's not a single optimizer that is best for all models. There are optimizers that are much better for specific types of models, but there are also different types. And so it's highly model specific. And this is why it's usually a common scenario that you do parameter estimation for a few steps with a few optimizers and then see who's the one that's best performing and then choose this one for the actual optimization. Which again means that you need to swap around a lot with the optimizers and you don't always want to adhere to the SciPy optimizer standard and how to do a parameter estimation there and then switch to the PySwarms optimizer because it requires a different data format. This is where pyPESTO comes into play because it interfaces a multitude of those optimizers and in addition has some in-house optimizers. This also directly allows for the easy comparison of optimizer performance, and you can additionally track the history because sometimes the optimizers don't allow you to do that, really wanting to check how fast do they converge and getting a better view of where we need to strike the balance. You can choose between local, global, and hybrid optimizers. I mentioned those already. well-known optimizers are probably the SciPy optimizers, all of them, and the LN optimizers. Okay, so how does it look like? Well, you only need to define your optimizer, which is one call if you do it right, with some options. In this case, we choose the SciPy optimizer, the LBFGS. We turn on the history and we can minimize. Just one function call. Obviously, I omitted a lot of options for the sake of fitting this into the slide. But we have fairly good default values, I would say, so you can really just start doing everything you want. And then down there, you can see a plot to compare the history of the optimizers. In this case, we compared LBFGS with feeders, and we can see, okay, how many optimizer steps did it take, and what's the actual value? And what does it show you? It shows you feeders converges much faster than the SciPy optimizer, but the SciPy optimizer finds a better value at the very end. And so now you got to strike the balance in how much computational demand can I allow, and how much do I want to compute, or is it much less important, this very small change here, and I choose the feeders optimizer to be faster. Gives you an option. This is just 10 starts. Usually you go with 100 to 1,000, and then you can't run it with the optimizer you like more. Okay, we are now at the crucial part, the visualization and seeing how well does the model fit. How do you do this? Usually it's through comparison directly, simulation versus data. How does it look like? We'll also introduce waterfall plots to allow you to get an estimate on the convergence, telling you whether you go back to step 2, step 3, something like this. Parameter distributions, which also help you in this, and some other options. All of them are, in the most easy case, one-liners. You can highly adapt them, but you don't need to. The first one is the model fit. If we look at this, for example, it shows you all the observables, and in the dotted line, it shows you the measurements, and the solid line is the simulation. We can see those two fit quite well. This one, we're not so sure whether we like this yet. And then we can have a look at the waterfall plot. So what is a waterfall plot? If we run a multi-start local optimization, we have multiple parameter estimates at the end. And so what we want to do is we order them by their objective function value, and then can basically get a feel on how often do we actually find our optimum. And so the more often we find our optimum, the happier we are, because we can assume that it might actually be the global optimum, because we only run local optimization. And in this case, for example, we see, okay, we actually find the best optimum quite a lot of times. We also find some local minima, apparently. And so the first question you can ask yourself, it's quite natural, is, do we actually always find the same point, or only the same objective value? And for this, you can turn to the estimated parameters. This shows you for all the parameters and all the starts, each start in the same colors in the waterfall plot, what is the actual best found value for the parameters. And you can see these are slightly different reds. I'm sorry for the choice of color here. The actual this red here is just one solid line, and it always finds the optimum. That's a good thing. A bad thing is it apparently always runs into this bound here. And so what we could do, for example, in this case, is go back to start 1, say, okay, we adjust the bounds, and perhaps we get a better fit in here. Just an option. But let's say we are fine with this. And we continue to determine parameter uncertainty. Again, parameter uncertainty means we want to calculate some kind of confidence interval for our optimized parameter to really know, is this the correct one? How certain are we about it? You've got a multitude of options here supported by pyPESTO. First one is the Fisher information approximation. You assume that your objective function landscape is normally distributed around your optimal point, and then you can estimate the distribution of the confidence interval by approximating it with the Fisher information or the Hessian, depending on what you provide. You can do Bayesian sampling, so Markov chain Monte Carlo sampling. pyPESTO supports a variety of samplers. PyMC4, I think, should be the most common one. It has 2 in-house samplers as well. The nice thing about Bayesian sampling is it gives you a holistic view. It doesn't only give you confidence intervals for one parameter and some only marginal distributions, but it actually gives you complete distributions. The downside is it really is very expensive and takes up to from 100,000 iterations to even more to get the Markov chains converging. It's slightly cheaper, but we only talk relatively, it's still expensive. Way is profile likelihoods, which estimates the marginal distributions of each parameter by fixing the parameter to a certain value and comparing it with the best found optimum then. And then by this, you know it's a chi-square distribution. So you have a distribution, you can get your confidence interval. Okay, how does this look like in pyPESTO? In pyPESTO, in here we have the example of sampling. You just choose a sampler and start sampling. So, straightforward. You can choose also another sampler and switch between them, and then you have some visualizations to show you, okay, how's the convergence of the chain? We can see here around 50,000 it starts to converge in this. How do the marginal distributions look like? You can also visualize the scatter distributions. We don't have them in here yet because there was no way to make them look nice on a tiny part of the slide, which shows you what is correlations between two parameters. And so with this, you can evaluate basically your confidence intervals and your uncertainty of the parameters. So the next step is we want to propagate this towards the prediction uncertainty. pyPESTO does this by generating ensembles from your previous results. Ensembles in this case means it's a collection of parameters that resemble basically the uncertainty in the parameter. And then you propagate this by simulating each set of these parameters and then looking at the output and how does the output vary. How can you generate those ensembles? You can generate them from sampling. Easy, you can just say, I'll show you in a second. I think for sampling I'll show you. You can also do it from optimization, or if you had it turned on from optimization history, determining, for example, I only want values that are in a certain range and have a quite good objective function value. I have to be transparent. This is the only one where I only show a partial code because it actually takes a bit more. The ensemble creation is, as you can see, fairly straightforward. Here we told this, okay, we want from this result where we did some sampling, every fifth value of the sample. What is a bit more involved is creating a predictor, creating a prediction result. Then we do have examples for this. So we have an extensive documentation in the Read the Docs. There will be a link for this later, but we don't have it in here yet because it, again, didn't fit the slide. Oh, no, just one click. Yes. So what you get out is prediction uncertainty. You can sort them as you like them because you either want to find out, do you have in a specific experiment uncertainties or do you have in a specific observable uncertainties? This one looks quite nice. So you'd be like, oh, I'm happy with this. Okay. So I think second to last step. Yeah, no, I think it's only the last step. Going a bit faster than I was estimating. Is model selection. Model selection is to determine model structure. Again, why would we want to do this? Usually it is the case that we don't know the model structure completely. This might be because there are a multitude of options already available, or even if there are only one option available, this option is not sufficient. So we have to create new options. If we have an uncertainty in the model structure, that means we need to choose some kind of best suitable model from the set of all possible models that we deem possible. That also means that we have to define what is the best model. And the way you do it is usually you have to strike a balance between complexity and goodness of fit. For this, you use usually the Archaico criterion or the BIC. both act in the same manner by giving a penalty on more parameters. A really nice example to just visualize this is if we take this example here, just 3 reactions. Let's say we turn off this parameter. So two models, either this one is 0 or it's not 0. If we assume it's not 0 and we estimate it, the estimation can just go very, very close to 0 and be basically the same model as if we had it turned to 0. So the fit and the optimization of the model will never be any worse than the one with less parameters. So you can add as many parameters as you want and you'll get a better fit. But you don't want to do this because at some point you'll just describe your data and not describe the mechanism. That's why those mechanisms are in there and penalizing more parameters. Okay. The way pyPESTO supports model selection is through ptab select. Ptab select is one of the extensions of ptab to, as it says in the name, describe model selection, also in a standardized format and with some tabular files. If we take this one here, for example, I already told you 3 reactions. How many possible models can we have? Well, each parameter that we have in there can either be 0 or it can be estimated, yielding us 8 parameters in the end, 8 models, 8 possibles. And so now the question is, how do we go through them? First idea would be, well, we could just brute force it, in which case this is going to be very expensive because in each estimation or in each model, we need to do a parameter estimation pipeline, which already was expensive, and we need to do this 8 times. And so there's a need to go through the parameters in some kind of algorithm, which is not trivial. There are forward searches, backward searches, basically telling you either add always a parameter, take one out. And there is an algorithm in ptab select that is supported that makes use of this forward backward and the fact that if a parameter value is much better with a more less complex model, you can exclude a lot of them, yielding you an exhaustive search without having to do all of the parameter estimations. One of the graphs that is shown, for example, in here is if we do the model selection, again, 3 calls, it's even easier than the creation of the problem, actually. It shows you how was the algorithm, how did it go through this, and what is the value of the AAC that it got. And you can see it starts out here with nothing was estimated, everything was 0, and this, by no surprise, is really bad. If we only add 1 parameter, it is really good already, and it doesn't get better if we add another one, but if we add all 3 of them, it gets much better. So in this scenario, it shows you what is the best values, and then you can decide basically which one of those you want. Okay, so coming back to the overview of parameter estimation, I hope I've shown you that pyPESTO streamlines parameter estimation. Obviously, we only went through each step separately. That's why I would really like to show you. This is everything from step 1 to step 4, leaving out the model selection and the prediction uncertainty, but model selection is like 3 more lines of code. And we can fit everything in this tiny screen, and half of it is importing the functions in the right way. And so by only having this, you can now switch between optimizers, switch between simulators, switch between samplers, change the options of the samplers, and really get the best out of all the tools that are available to you. And in this manner, pyPESTO allows parameter estimation for a wide variety of models. There's a vast amount of additional options that I haven't shown you here. One of the examples is that we try to parallelize this to make it highly computationally efficient to be able to use it on clusters, because simulation is usually so expensive. You don't want to have your computer run all day because you can't do anything else. It takes up all your CPU. So you run it on clusters and it's optimized to be used on this. We have an extensive documentation in case you're saying now, okay, I'm interested, but I really don't know how to use it. click on the link, and there are a lot of examples for most of the applications that we have and most of the features. You can also directly check out the GitHub with this cool QR code, and don't worry, the QR code will stay on the next slide, which is the thank you slide. I promised you one in the very beginning, and it is here in the end. It consists also of a functional overview of what I didn't describe yet, because one of the few things is parallelization we already talked about. Storage is something we need to do, because if you run it on a cluster, you don't want to do the interactive visualization part. We save it. You have hierarchical optimization, which tries to reduce the dimensionality of the parameter estimation problem. And we can handle more than just quantitative data. We also have support for semi-quantitative data, qualitative data even, and even the option for more complex observation functions. Many thanks go out to my PI, Jan Hasauer, for allowing me to do my PhD, my co-authors of the paper, and I already said all the pyPESTO developers keeping the tool alive. I think everybody who has a tool knows that it's always a pain to keep it alive, and it's always a struggle. There are some tools that we have that didn't survive, to be honest. And the whole Hasenauer lab for providing me with an environment to do my PhD, which I'm very happy about. And last but not least, the Sauro lab for giving me the possibility to have a talk here, and as Merkel already mentioned, having me over in March to integrate Roadrunner into pyPESTO. There's a QR code for the paper and a QR code for the GitHub. Thank you for your attention, and I'm happy to receive questions. I was shooting for 40 minutes. I'm just saying I did. Okay.
That's fantastic. Thank you, Paul. Yeah, open to questions.
I think there are some in the chats. I take a second for reading two of them.
Yeah, one of them was mine. Although Bill replied, I mean, we've done a lot of, on another project, we've done a lot of this sampling, and it takes forever to converge. So if you use one of these optimizers first and then do the sampling, I imagine it'd be much faster.
Yes, that was the idea, basically.
Yeah.
That's why we do the optimization first in the pipeline, because otherwise, you're right, you can also just start randomly. Another thing we have is parallel tempering, which allows you to jump. So you have a multitude of chains, and they exchange by having different values for the probabilities to jump in between optima. And so if they exchange information, the possibility of going out of local optima, because that's mostly what hinders you, is increased.
Yeah, right.
Yeah, I think so. How well and to what extent do you tolerate very partial observability? I was asked. Partial observability in the sense that we have data that is only measured up to a certain amount or below a certain amount. Usually it is the question of how do you propagate this to the distribution. There are a multitude of options that depends on the censoring of the data. But pyPESTO is able to handle this. So mainly I would have to say, to be fair, I think it's ptub that is able to handle this. And then pyPESTO uses this by just saying, okay, if it's below a certain amount, then there's a certain distribution for it. The likelihood function changes. But we are able to do it.
There's a question from Joe.
I'll let you read it.
Oh, you let me read it. Oh, there's another question.
Okay, yeah, okay, I'll explain it. Yeah, so, I mean, obviously, the computational demands are huge, and especially for biological problems where, you can have 60, easily 60 parameters in a modest model. So yes. I was just curious in terms of, what strategies might you use in terms of selecting subsets of parameters, maybe to get, because the whole trick to this is to try and get close to something so that then you can use techniques that have a chance of finding, a decent solution. So I mean, it's a combination of maybe selecting parameter subsets. That's one way that you can reduce the space size. But obviously there are interdependencies here with the other parameters, so that's a challenge. And the other challenge is, the range of parameter values. Because again, in biology, parameters range from, depending on the units, you can get things that are fractions of a unit to millions of units. And so so that's another one in terms of an optimizer, which is hugely challenging. You don't want to go, fractions of units increments, or explorations for something that can go up to millions. Oh, I just wondered what strategies, what strategies you use to try, because this is something under the user's control that they can play with. Yeah.
So we've played around with some options. So one thing I have to mention first is usually even like stopping early or things like this are usually up to the optimizer. And this is where we don't play the part because we interface to the optimizer, but choosing a good starting value for this is, as you already mentioned, determining good bounds for this, determining how do we want to start the actual optimization. So one thing we played around with is just randomly sampling starting points and looking what are good starting points. Multi-start local optimization works really well with this, for example, but you can also do it without directly starting the optimization, just looking what do you think is a good area, though one has to keep in mind then your optimization kind of turns biased. This is also something you always have to keep in mind. You don't want to be too biased. Another thing that really helps, especially in high dimension, two things, is gradient-based optimization. We found that gradient-based optimization really speeds up the optimization, and then it comes down to more efficiency in the simulation, because independent of how many parameters you have, independent of how big the problem is, and mostly even independent of the optimizer, I'd say the thing that costs the most is the simulation. And so that's also something out of our range because we only interface the simulator. But I think Roadrunner and Amici do a very, very good job in being very efficient in the simulation. And the last thing to reduce dimensionality is the hierarchical optimization I just briefly mentioned, which reduces the dimensionality space by realizing that parameters that you want to estimate that are based on the uncertainties, like you want to estimate the variance, for example, of your noise, or the scaling parameters, they can be taken out of the whole loop of parameter estimation, and you can have an analytical solution in each step. And so you decrease the dimensionality of the problem by how many states you have. Okay, I'll take a second. I hope that answered the question. I'll need to read through the other questions for a second. I was asked whether pyPESTO has a built-in tool to track number of objective function calls during sampling. Yes. Though no. Yes and no. Because you define in the sampling process how many samples you want to do. And usually that is actually the amount of objective function calls you do. Because one sample is one objective function call and evaluating it. I hope that helps. So it isn't defined in the result, but you already have it defined when you say how many samples do you want to take. Yeah. I hope that answers the question.
Can I just follow up with that?
Yes, yeah.
Okay, thanks for answering. My question is super specific, but so I don't think that's necessarily true, because I looked at the source code and I tracked how when exactly the function is the objective function is actually called, because you can compute the prior first, right? And
Mhm.
the thing is like if the prior is invalid, the objective function isn't called, but I don't know if that's counted in a step. So like I wish there was just a way to directly access when the objective function is called or not, because some steps there might be an invalid step and then it rejects it, so the objective function is never called, but that would be counted in terms of the number of iterations, right? So yeah, do you see my problem?
Yeah, I see the problem. I would think that there should be a way around this, because if it is counted up, it should be, but I'm just guessing from what I know about the source code. I don't know by heart, I'll be honest. You probably know more about it if you looked at it very recently in the sampling case for the objective evaluation. But you should be able to see, because it should store like an NRN value or an infinite value in the chain. And so if you remove those from the chain, you might get it. But no guarantee for you there. It's a good thing. We'll have a look into it. I'll open an issue, and either we can answer this or we open a pull request to be able to do it, because you're right, it is important. Yeah.
Okay, I could actually upload the issue and like give you exactly like what I'm looking for, so I can like continue this conversation, but thank you.
Oh yeah, perfect. That's even better. Thank you. Okay, have I missed another question? Oh yeah, there was another follow-up question on the partial observability on discontinuities or lack of smoothness, and I'm assuming lack of smoothness in the objective function. If it is in the objective function, it is honestly not up to us, because then it would again be the simulator that simulates it. If you have really discontinuities in the sensitivities, then you might want to do a sensitivity-free optimization. This is also possible using some other optimizers. That's the nice thing about pyPESTO, being able to choose between the optimizers you want to do, depending on what is available to you. I hope that answers that question. Yeah, then we have any more questions?
I guess I got one question. I haven't used pyPESTO, so I may not know what I'm talking about. But if you wanted to do a really simple problem, do you have to specify the objective function, or is there one already built in? I mean, you know, I guess you have to give something, I suppose, difference between, yeah, you have to specify some objective function, I guess.
Yeah, but
There's
I would
nothing
say
built in.
if you just want to run an example or something,
Yeah.
then it's just loading some PTAB file from, we have a benchmark collection collecting, I think it's currently about 30 or 40, I might be lying there. I think it's somewhere around the range of models you can just take in, use them, and have a look. We do have a function in the tests that creates you some kind of values and some kind of objective functions, but I'm assuming that what you mean is I have some kind of model and really want for this model an objective function that is easily done.
Yeah, I mean, I'm just wondering how easy it is to start up using pyPESTO. If we gave it to somebody new, how much stuff how much boilerplate stuff you have to write before you can get something running.
I would say not too much, because if you want to do parameter estimation and you want to do it sufficiently sophisticated, you need to think about what parameters do you want to estimate and what are the things that we measure. And if you think about all those steps, then you basically have created a PTAB problem already. Because in the easiest case, if you just want to use an example, just use the identity function, meaning you put in all observables, just the identity, you use some kind of normal additive norm distributed noise. There are options in PTAB to take from the data table and the condition table to just directly create and together with the SPML model, create the parameter table. So there's also, you don't need to do
Do you have a really simple
So
example available? I'm trying to look through the
uh,
docs. I don't see anything yet. Like, you had that the cell curve model at the beginning. Let's say you wanted to fit all parameters and you had, was it 2 variables? Both variables
yes.
as quote unquote
Let
experimental
me
data.
let me quickly go through this to the p-type definition, and then I'll try to tell you. I'm for now omitting the measurement table because that depends on how much data I have. Because without data, it's one line for each data I need to add. The observable table would just be 2 lines. Parameter estimation table would be 5 lines. And I have 3 conditions. So
Oh, I see.
it's actually parameter table would be actually only 4 line because the K2, the parameter, would be in the condition and it would vary through the condition. And so I think it's in total 2, 4.
But if you wanted to fit all parameters, you'll just have that many parameters in the table, in the parameter table, right?
Yes, then it would be
Okay.
5 parameters. Then the condition table would be just 1 condition.
Condition table is 1 condition. What does the
Yeah, it
condition
would be-
table indicate? What does that indicate?
It's telling you if you have different conditions, experimental data sets, basically. That
Oh,
are
right. Okay, let's
performed
say we
under
only
different
have
conditions.
okay, let's say we only have 1 time series for 1 time series
Yeah,
for
then the
each
condition table
variable.
is it cannot be empty. So it says
Yeah.
condition ID, condition 1. That's the whole condition table. You get rid
Yeah.
of this. The measurement table again is as many lines as you have data points. The observable table is 2 rows, and the
Oh, I
parameter
see.
table is 5 rows. And so that's it. So
I think if you have an example that just say fits the cell curve model, you try to fit all the parameters and you have 1 set of time series data for each variable. That would be a nice example. Because I'm going to be doing a class in the fall. Be nice to show them
I'll happily
simplest
send.
possible
I'll happily send you
example.
a very simple problem. Yes, no, I can
Oh,
definitely share
Caroline.
one with you.
Caroline says she
Caroline has one.
oh, that'd be nice. Yes, Michaelis Benton is a
Yeah.
good one.
Yeah.
I can email it to you. It addresses exactly what you're looking for, which is it just has a single condition.
Yep.
And it fits to like synthetically generated
Right.
trajectory of the product.
That's what I'm after. Yes. Oh, that'd be
Okay.
great. Yes.
Sure thing.
Thank you.
Thank you, Caroline.
Yeah.
I can also, Herbert, send you the link to the benchmark collection because there, then if you want slightly more complex,
To go beyond,
depending
yeah.
on, you can have
Right.
all of them. And there's simulation data or there's actually explicit data in there already.
Okay.
Depends on
And installing on Windows is literally just pip install pyPESTO, is it? And that's it.
It is on Windows. If you don't want to use Armici, but that's Armici on Windows, that's a different thing. But since we support Roadrunner, it should be just
Yeah, I
pip
don't have a
install.
compiler. The students won't have a compiler on their machine.
Yeah, it should be just pip install pyPESTO.
Okay.
I'll check again.
Yeah.
Because
Wait, sorry,
yeah.
I was just gonna say, like, my understanding right now is that currently, like, the lib Roadrunner pipeline where you like import PE tab with Roadrunner, that's not decoupled from Amici. So you still have to have Amici installed in order. So I work on a Windows machine, and my workaround is I just use the Windows subsystem for Linux, and that's worked.
Okay.
I haven't decoupled that. That is a good thing. I should look.
Yeah,
Perhaps because of the PTUP importer. Because the PTUP importer probably requires Amici. Is that the case?
Right. It's just in like the imports at the top of the file. Yeah.
That's a good observation, Caroline.
Thank you.
Yes, we'd like that decoupled if possible, because the students won't have a chance. They won't be able to go into a subsystem like you can, Caroline. They'll be completely lost.
We will be able to decouple that.
Decouple it. Yeah, our class starts in October, so if you have anything that you have a decoupler, that'd be great.
I would have said probably in the next week we can do that because
Okay.
it should really just be about the import. I mean, that's why we designed Roadrunner as it is to be
Yeah,
decoupled from Armagy.
we can't. Yeah,
Yeah.
I think Joe Hellerstein may have had the same issue actually, that he noticed there was a coupling and he couldn't get it to run. So
Yeah. We'll get around that. In the
Okay,
worst
great.
case, it's just an if not import or if import error, just
Right,
say none because it shouldn't be used anywhere
that'd be good.
in
That'd
the
be good.
PTAB import for the
Right.
Roadrunner.
Thanks.
Yeah, thank you for pointing that out. I wasn't aware because I have Armagy installed, so it never turns up. And I think
Yeah.
in the test it also doesn't turn up, but it might be because that's also Linux again, and then it's easier to install.
Yeah.
That's it. It's
Okay,
important to
good.
know. Yeah. Okay.
All right, yeah, we're at 4:06, so thank you, Paul, again for your time.
Thank
And
you for
yeah,
having me.
I encourage everybody to go back and watch, this seminar and previous seminars from the website. These will be up shortly. So thank you. And with that,
Should I send you
we'll,
the slides as well? Do you upload the slides as well? I can send them to you if you want to.
Oh, everything that you included in the seminar will actually be on the YouTube video.
Ah, okay. Yeah,
So
good.
I don't think you have to set that stuff.
Good.
Okay,
All right.
thanks a lot.
Thanks again.
Bye now.
Bye.
Bye.